Grain Research Program is a recently founded program that was formed by a group of multidisciplinary researchers from different institutes across South Africa and from programs at FABI. This program was developed with the purpose of engaging with grain stakeholders and also to conduct research that will support the grain sector and in turn contribute to South Africa's food security and bioeconomy. One of the many threats to grain production in South Africa is the invasion of Lepidopteran pests, which feed on and destroy many economically important crops, including maize and sorghum. At FABI, critical research is being conducted on these pests so that the populations can be adequately controlled in South Africa. The fall armyworm is a lepidopteran pest which feeds on many economically important crops, including maize, sorghum, rice, sugarcane, and cotton, just to name a few. The fall armyworm is indigenous to North and South America. However, it became invasive to Africa in 2016, and since then it has rapidly spread across sub-Saharan Africa, including South Africa, where it was first reported in 2017. And I'm currently doing a master's degree in genetics at FABI. My project focuses on understanding the population genetics of the fall armyworm in South Africa. By analyzing the DNA of fall armyworm samples in various populations in South Africa, we can then compare their genetic makeup between individuals and between populations. We can then understand the gene flow between different populations, and in doing so determine the migratory pathways of this pest in the country. Because information regarding the number of populations present in South Africa and the degree of connectivity between these populations is critical for developing robust management strategies to control the pest. Using population genetics data, we can also look into the groupings of alleles within populations, and by comparing this data over time, we can identify whether there have been any contractions in the genetic makeup of that population, and this can reflect on the efficacy of the control strategy that's been applied to that population in question.
My name is Maize. I am a PhD student and I'm working on a pest known as Busyola fusca. Busyola fusca is commonly known as the African maize stalk borer, and it's a pest of maize and sorghum. This pest is very problematic as it tunnels through the stems of the crop, creating holes, dead hearts, and also destroying the crop in the process. The populations of this pest are managed using transgenic maize expressing pesticidal genes known as the cry genes. The maize expressing these genes is known as Bt maize. In the past 14 years, unfortunately, this pest has developed resistance to Bt maize. My research is therefore focused on understanding the genetic basis of Bt resistance in this pest. I will be looking at the genetic variation between susceptible populations and resistant populations of the pest. I will also be using a genome editing approach in order to look at specific genes that may be involved in Bt resistance of this pest.
At the FABI Biocontrol Center, we've established a rearing colony of these pests, which includes colonies of both resistant and susceptible populations. A large component of this process involves rearing insects on an artificial diet based on dried maize leaves. And this diet needs to be prepared on a regular basis. The larvae of these pests are reared solely on the artificial diet in which they pupate. Following this stage, the pupae are then transferred to a breeding cage that contains live maize plants, where the adults will then emerge, mate, and lay their eggs directly onto the plants. The neonate larvae that hatch from these eggs are then placed back onto the artificial diet, and the cycle will repeat itself.
I grew up in a village where maize is not only a source of food for the household, but it's also a source of income for resource-poor farmers. So I'm really proud to be actively contributing to research that is going to contribute greatly to food security in my country.